Good morning. This is the first lecture in the Stevens Scientific Services lecture series. And today I'm going to focus on the pathogenesis of the coronavirus known as COVID-19. I'm a professor emeritus at Harvard Medical School. I was there for 36 years. If at the end of this talk, if you have questions about anything I had to say today, please feel free to send me an email. My email address is rstevens at richardstevensphd.org. If you'd like to know more about my research over the years, please go to my website, www.richardstevensphd.org. And more recently, my wife and I have created a new business. It's a consulting business. It's called Stevens Scientific Service. If you'd like to know more about that business, please go to that site, StevensScientificServices.com. I'm taping these talks, and all the talks will be available at the Stevens Scientific Service website. Now, as a disclaimer, I'm a PhD, not an MD, so thus any questions that you might have that are clinical nature, I recommend that you talk to your clinician. So my talk today is going to focus on coronaviruses. And coronavirus is so named because they have this crown-like feature on the outside surface. And here is an electron micrograph of 5 COVID-19 viral particles. And on the right is a model created by the Center for Disease Control, which I'm sure many of you have seen. And these crown-like features are represented by these red uh, uh, parts on the model, and these are called the spike proteins. And this is the part of the molecule all the vaccines are directed against, because this is the part of the molecule that the virus uses to infect human cells. Now, coronaviruses are very small. They're only 80 nanometers in size, and they're even smaller than the HIV virus. And put it in size in perspective, they're about 125-fold smaller than the E. coli bacteria in your gastrointestinal tract. Now, there are many types of coronaviruses. Some only affect animals, but there are 7 that have been identified over the years that affect humans. And the 3 I'm going to focus on are SARS, MERS, and COVID-19. And these 3 coronaviruses are similar in that they all 3 induce a severe acute respiratory syndrome. And indeed, that's how the name SARS first appeared. Now, SARS was identified in China in 2003, and there was 1,800 people became infected, and about 800 of them died. And while it's a 10% death rate, it's 100% fold worse than what occurs with seasonal influenza. Now, MERS appeared in Saudi Arabia in 2012, and it had a mortality rate of 35%. Now, not many died of SARS or MERS relative to COVID-19. And the reason for that is because the spread of the first 2 coronavirus was quickly contained. Well, COVID-19 is another coronavirus that affects the lung. It first appeared in China in 2019. And that's the reason why it's called COVID-19. It isn't the 19th coronavirus, as one of President Trump's advisors concluded. It's just because it appeared in 2019. Now, COVID-19, we're fortunate, and it's less lethal. Its mortality rate is about 3%. Nevertheless, it's more infectious. And that's the reason why there's essentially no place in the world today that is COVID-19 free. Now, RNA viruses mutate to evade our immune system. They're trying to survive. So as we attack the virus mutates to try to survive. And there's already strains of COVID-19 have been identified that are more infectious. There's a strain in the UK, South Africa, Brazil, and more recently, California. And whether these new strains are more lethal than China's COVID-19 and whether one or more of them affects children remains to be determined. Now, in my next talk, I'm going to focus on the adaptive immunity to COVID-19. And for those who are interested in COVID-19 immunity, the rate of mutation of a virus impacts one's ability to create a vaccine that is effective and long-lasting. Its high rate of mutation is the primary reason why no HIV vaccine has been developed, despite this retrovirus appearing in the 1980s. Now, fortunately, COVID-19 isn't mutating as fast as HIV-1. Now, on this slide is a model of the outer surface of COVID-19 and a cutaway of what is going on in its surface. Now, all the action, all the antibodies that are being generated by the various vaccines are attacking this protein on the cell surface known as the spike protein. And the reason for this is twofold. One is because of the outer surface of the virus, and two is that this is the part of the virus that uses to infect human cells. Now, it turned out that SARS and COVID-19 were separated by 16 years. SARS was 16 years ago. And both of those coronavirus have this spike protein. Now, even though it was 16 years separated, the spike proteins are 76% identical at the amino acid sequence level. And in addition, there are numerous regions in these spike proteins of SARS and COVID-19 that are 100% identical, as I'll show you in a bit. Now, that information can be used to generate COVID-19 specific antibodies, as well as antibodies that recognize both coronaviruses and probably new strains of COVID-19. Now, it just took 3 months before the genome of COVID-19 was determined. And the reason for that is it's an RNA virus, and the genome is quite small. It's only about 30 kilobase, and that's less than 0.1% the size of the human genome. And the genome encodes 29 proteins. The protein we're most interested in, the spike protein shown here. And that spike protein when it gets translated, it's 1,273 amino acids in size. And the protein has two domains. It's called the S1 domain and the S2 domain. The S1 domain is the part of the spike protein that binds to human epithelial cells via the receptor called angiotensin converting enzyme number 2. And the spike 2 subunit is critical for the uptake of the virus inside human cells. So here's a cartoon explaining how COVID-19, or it's also known as SARS-CoV-2, attacks and infects human cells. So the virus is floating in our system, comes in contact with the human epithelial cell. The human epithelial cell has the receptor on the cell surface called angiotensin converting enzyme number 2. The virus binds tightly. It undergoes a conformational change. And then when that occurs, the second step is this protease called transmembrane serine protease number 2. Is a triptych type protease that will cut between the S1 and S2 domains, allowing the internalization of the virus into the human cell. And this cartoon depicts on what's going on inside the infected cell. So once again, here is COVID-19. It approaches the human epithelial cell. It recognizes the receptor ACE2. Then the TMPR SS2 nicks it. When that occurs, the virus gets taken up by the cell in a vesicle called the endocytic vesicle. That's a lipid vesicle. Once it gets into the cytoplasm, it releases its cargo. The genome, the RNA genome of the virus gets replicated. You get many copies of that. And then also the RNA gets transcribed, translated. And basically what happens is you take over the machinery of the human epithelial cell. So the human epithelial cell may make hundreds and hundreds of mammalian proteins, but that's completely shut down at the expense of producing viral protein. Viral proteins assemble. They get put into this lipid vesicle. That vesicle fuses with the plasma membrane and releases the viral particles. Now it was very quickly discovered that angiotensin converting enzyme is a primary receptor for the coronaviruses. And because of that, it thought, well, maybe we can develop a pharmaceutical approach where we can actually block ACE2. And if we do that, we will prevent COVID-19 infections. And despite that initial hope, the ACE2 inhibitors can't be used to block COVID infection. And the reason for that is because ACE2 is critical in catabolism of this peptide hormone known as angiotensin 2 or angio 1 to 8. So this is a hormone that has a tremendous bioactivity in our body. The precursor hormone is called angiotensinogen. It's 485 amino acids. That's made in the liver. That gets secreted into our blood. It makes it to the kidney, and there's another enzyme there called renin that clips this big protein, liberating a 10 amino acid peptide called angiotensin 1 or angio 1 to 10. That peptide, if it makes it to the lungs metabolize it again, and you get a 8 amino acid peptide called angiotensin 2 or angio 1 to 8. And this has potent bioactivity in the body. It regulates bloods, vessels, it regulates kidneys, it regulates heart, adrenal gland, and brain. And so this is a dangerous hormone. So we must control the level of this hormone, not only by producing it, but also by degrading it. And the enzyme that's critical in degrading this is angiotensin converting enzyme number 2. It takes this angio 1 to 8 to 1 to 7. So thus, if you block angiotensin converting enzyme 2 in an attempt to save ourselves from a COVID-19 infection, you might actually have more damage because you will not be controlling the level of this peptide. So how that's done was it became very clear early on that we need to develop vaccines. And in my second talk, it's going to be focused on vaccine development, and that's called adaptive immunity to COVID-19. Now, I'd like to spend a few minutes talking about the symptoms of those who are affected by this virus. Common symptoms, about 90% of people develop fever, about 70% have a dry cough, and about 40% have fatigue. But many other people have other serious problems. Headache is common. Loss of smell, loss of taste is common. Nasal congestion, sore throat, coughing up sputum, shortness of breath, pain in the muscles, chills, nausea, and even diarrhea. About 30% of the people will have diarrhea. Now, this is the mild case of the disease that can progress to the severe case. In severe case, you get is life-threatening, difficult walking, confusion, coughing up blood, decreased white blood cell, kidney failure, high fever, but those who really go into the hospital often require oxygen, and they experience internal blood clots as well as a cytokine-chemokine storm, and that's induced by some inflammatory mechanism which remains to be determined. Now, depending on one's age and other factors, up to 20% of COVID-19 cases progress to severe disease. Now, the organ that's most studied is the human lung, and due to the inflammatory response to the pathogen, fluid accumulates in the lungs of those who require hospitalization. The lungs become scarred, less elastic, breathing is impaired, and to the point where oxygen becomes dangerously low, and these patients have to be put on oxygen ventilators to increase their oxygen content. The most severe cases, your immune system goes into overdrive, sometimes at the expense of other organ, and this is called a cytokine-chemokine storm. And the mechanism hasn't been identified. Having said that, I think it's mast cell dependent. Now, as the body attempts to fight off COVID-19 infection, the lungs become more susceptible to secondary pathogens, for example, pneumonia, which can lead to sepsis, as apparently happened to the radiotoxicology Larry King. Now, Larry King got COVID-19 infected, but it was reported that he died not of the infection, but of the pneumonia. So what is it? Another big problem in the lungs is that you develop these light-threatening blood clots, and that you do not want to get a blood clot. The blood clots are number 1 and number 2 cause of death in the world in most years, leading to heart disease and stroke. So this is the lung, but please note that COVID-19 can also adversely affect the heart, the kidney, the gastrointestinal tract, and the nervous system. Now, shown on this slide are three X-rays taken by Dr. Bankhead Kendall at the Texas Tech University. And what she did is that she looked she did X-rays of normal, the lungs of normal, healthy people, those who had advanced COPD due to their chronic smoking cigarettes, and then the lungs of her COVID patient. And what you see is that those who progress from healthy to COPD, you get scarring and damage. That's cloudy areas, and and the lungs collapse. You get fibrin platelet clots that are also present, and shortness of breath lingers even after you live in the hospital. So this is the lungs of one of her COVID-19 patient. So even if this patient recovered and was able to survive the life-threatening pathogen, uh, you've already damaged the lung. And so it's going to be the next 20 years looking at the long-term consequences of COVID-19 in the human body. So 500,000 people died, but millions of people or have damage in different parts of their body. Now, on this slide, this is data taken from the World Health Organization as of 9 January last month. And so the World Health Organization looked at this period from March to January, and they identified 88 million COVID-infected cases in 220 countries, and that resulted in about 2 million deaths. Since about 50 million people recovered, the overall, to some extent anyway, the overall mortality rate is presently about 3%. Now, having said that, mortality increases dramatically up to 15% if an infected person who is very sick wasn't treated in the hospital. And that indeed occurred in Italy and Spain. So here is the death curves of Italy in greens, UK in purple, Spain in red, United States in dotted blue, Sweden in black, Canada in dotted red, Germany in yellow, and South Korea here in brown. So what happened in Italy and Spain is that so many people got infected early on that the hospitals could not take care of all those people that it normally would, and so the death rate was about 15%. Then Italy and Spain and other countries Sweden and Germany, what they did is to close the country down, and then the death rate did not increase. Then when they opened up the country, all of a sudden the deaths started increasing. Now, the United States never plateaued out like Spain or Italy or Germany, and the reason why we never shut the country down due to the policies of President Trump. Now, some countries did a really good job. So it is possible to control this virus. So you can see Germany did a much better job than Italy, United Kingdom, and even the United States. The best country was South Korea. And the same day the first case of COVID-19 was identified in South Korea was the same day in the United States. And you can see the dramatic difference in the ability of the United States to combat the pathogen relative to South Korea. And the reason for that was that SARS appeared in 2003. SARS started to spread to South Korea. South Korea knew the danger of coronaviruses very early on, so they were very tough in that first couple weeks of testing and making people wear face masks. So the bottom line, this is South Korea educated the world. This is what we need to do if this ever occurs again. Now this table puts the perspective of COVID-19 in the big picture of American deaths. So as of February 26th, there were 510 confirmed cases of death of COVID-19. And it's now varies the death rate between 2,000 to 3,000 a day. But if we say conservatively 2,000 a day, let's say that the pandemic lasts another 2 months at that rate, that's predicted to kill another 180,000. And if we add that to the 510, we'll get 690,000 deaths. And the number of deaths, even a conservative estimate, is going to be the most that we ever experienced. The Spanish flu was a terrible pandemic in 1918 that killed 675,000 Americans. The Civil War, 630. So this is going to be the worst. Now, most years, ischemic heart disease is the number one, stroke number two, and COVID number three, leading causes of death in the United States. In 2020, COVID-19 was the second leading cause of death, just behind heart disease. Now, there's a lot of talk about herd immunity, and so I'd like to clear that up. So herd immunity occurs when a large percent of the human population becomes immune to a pathogen. If enough of the resulting survivors are no longer susceptible to the pathogen, it has nowhere to go. And natural herd immunity eventually occurred in Black Death, bubonic plague pandemics in Europe in the 1300s. So if that pandemic 1 year killed 50%, and then the next year killed another 50%, after 3 or 4 times, the survivors become resistant to the pathogen. Now, the chief White House advisor for President Trump was this man, Dr. Scott Atlas, and he advocated it last year that the Trump administration do not attempt to stop the spread of COVID-19. And his approach, natural herd immunity, in contrast to vaccine herd immunity, COVID-19 would eliminate the elderly and those with pre-existing health conditions and people of color. And the premise of this cold-blooded approach is that the resulting deaths are simply the price America pays to prevent collapse of the economy. In his mind, COVID-19 simply is nature's way of thinning out the human population by removing the oldest and the weakest. Now, the central flaw in his approach is the price that we would pay. And even with use of oxygen ventilators in ICUs, the COVID-19 mortality rate in the United States has been 3%. Now, if the White House decided to do nothing to stop the spread of COVID, as Dr. Atlas proposed, at least 10 million Americans would have died. And you get that number by multiplying 3% mortality rate by 325 million Americans, and that gives you 9.8 million deaths. But in reality, 30 to 50 million Americans would have died. And the reason for that is America's hospitals would have been overwhelmed, thereby increasing the mortality rate from 3% to 10 to 15. And indeed, this is what happened in Italy and Spain. Now, fortunately, most concluded that Dr. Atlas's natural herd immunity program was not worth the anticipated number of lives that would be lost, even though it made sense economically to President Trump. Now I would like to spend a few minutes going over the major risk factors that will determine whether you live or survive when you're infected with COVID-19. Now, COVID-19 disease symptoms vary widely. Many have no symptoms, whereas others became so sick they eventually need to be on an oxygen ventilator in the ICU unit just to breathe. And then there's that 3% who actually die. Now, mortality is higher in males and females. In China, the initial death rate in men was 4.7% compared to 2.8% in women. Now, in Italy, in the first wave of the infection, when the hospital was overwhelmed, it was 16.6% in men compared to 9% in women. Now, the risk of developing dangerous symptoms is also increased in people who are older and have serious pre-existing health conditions, what I'm going to go over in the next couple of minutes. Now, while each of these problems increases the risk of life-threatening COVID symptoms, those with one or more are even at higher risk. So let's go through a few of the risk factors. The first one should be pretty much obvious to everybody now: not washing one's hands, not wearing a face mask, and not maintaining social distance of at least 6 feet. Now, regarding the washing hands, it's been discovered that COVID-19 can live up to 3 days on a stainless steel or plastic surface. So thus, if there was a person who was highly COVID-19 infected, and they coughed and those viral particles landed on a bench, and then you put the hand on that bench, you would pick that virus up. And if you touched your mouth, you would infect yourself. Now, regarding face masks, they protect the wearers and also those around them. And the thing that drove me crazy is that it'd been known for more than 100 years of the importance of face masks. And this is a picture taken from the New York Times in 1918, showing the police officer. And note that every single one of the police officers had a face mask. And as I said, South Korea, in the United States, the first case of COVID-19 played on the same day. But South Korea has almost no deaths. And the reason for that is because they knew right from day one the importance of wearing face masks. The importance of face masks and distance is shown quite dramatically on this picture. Now, this is a man who is sneezing. This is from Wikipedia, and you get these viral respiratory droplets that are excluded out of his mouth. They vary between 5 to 10 micrometers in size. Now, Sender and co-workers looked at a highly infectious person, and he and his lab concluded that each infected person had 1 to 100 billion COVID-19 viral particles. And if that's correct, an individual who ingests or inhales a COVID-19 respiratory virus can be exposed to millions of these viral particles. Now, N95 masks are highly effective because they can block the uptake of greater than 99% of COVID-19 viruses. Now, having said that, face masks can be highly effective if they used properly. And this is the Center for Disease Control guidelines for wearing face masks. So you should choose masks that have 2 or more layers of washable, breathable fabric. And the washable is good because after you wear the mask one time, you can put it in the washing machine and kill the virus. You need to use a mask that covers both your mouth and the nose. I see many people with a face mask over the mouth, but they don't have it over the nose. And so the virus just goes into your nose and infects you that way. In the same way, it's not only the nose and the mouth, you also need to get tight fittings on the side. Now, what you don't want to do is you don't want to wear a mask that's hard to breathe, such as some of the vinyl masks. And you also want to avoid these polyester neck masks. And the reason because they're too porous and too much virus can escape. There are people who have these masks that have vents on the surface. And if you're going to use those masks, you have to have an inner cloth filter to block. And lastly, don't use masks that are intended for healthcare workers. And indeed, the probable reason why Dr. Fauci and the White House Task Force did not advocate wearing face masks in the first month or so of the pandemic is because they were worried that there'd be a run on N95 mask and there would be enough for the healthcare workers. The second factor is genetics. And then this remains to be determined, but what's now clear is that Blacks and Hispanics, if you're a COVID-19 infected Blacks and Hispanic, they're 2- and 3-fold more likely to die of the pathogen than similarly affected whites and Asians, respectively. And the worst are Native American Indians and Alaskans. And so we don't know why that Blacks, Hispanics, and Native Americans, Indians, and Alaskans are susceptible, but it might be due to mutations in the ACE2 gene and the TMPRSS2 gene. So people are starting to look at this now, and here is one study, and they looked at 9 different regions of the ACE2 gene. These are called SNPs, or single nucleotide polymorphism. And they found that there was, in the Asians who are fairly resistant to the pathogen, they have this particular SNP, whereas those who are Black have this SNP, and those who are Native American have this. And so the idea is that if you have a point mutation in your ACE2 gene, you will either be more susceptible or less susceptible to the pathogen. The third major risk factor is age, and about 80% of the COVID-19 induced deaths in the United States occurred with people who are 65 or over. And the risks are even higher for the elderly who have underlying health conditions or are living in a nursing home. And one of the reasons for this is that the pathogen can spread easily between people who live in close proximity to one another, especially indoors. Now, this is Center for Disease Control data taken a few months ago. It needs to be updated, but I think it's probably still quite valid. So what the Center for Disease Control looked at is different age people. They didn't segregate them by whether they had a pre-existing condition, whether they were male or female, whether they're black, white, or Caucasian, just their age. And then they looked at these people who differed in age. What was their hospitalization rate and what was their death rate? And so they had to pick a age bracket for a comparison group. So they picked these college age people, these 18 to 29-year-old people. And note that as you increase, it becomes a full increase of going into hospital if you're infected. So if you're 65 years old, you have a 5-fold increase of susceptibility of going into the hospital if you're infected compared to a 20-year-old. But really where the dramatic difference is the death rate. So by the time you're 65 years old, if you're infected and you're going to that hospital, you have a 90-fold increased chance of never coming out of the hospital. Now, the data gets even more dramatic if you compare it to this preteen group. So this is 16-fold lower than the death rate is 16-fold lower in this preteen to teen group than the comparison group. So if you now compare 65 years old to the preteens, you're actually 1,440 times greater. And the situation gets worse. If you're a man, double this by 2. If you're a black, double it by 2. If you have a pre-existing condition, double it by at least 2. And so this is the reason why vaccination of the elderly is so important. A fourth reason is that the lung is affected, as you notice that many of the people who are in hospital are on ventilator. So if you already have a pre-existing lung condition like COPD, lung cancer, lung cystic fibrosis, idiopathic pulmonary fibrosis, or even moderate to severe asthma, you're going to be increased risk of developing COVID. So these people, the people who have COPD, for example, need to really take precautions that they don't become COVID-19 infected. The fifth risk factor is those who have heart disease, diabetes, and obesity. And the reason for this will become apparent at the end of my talk, which namely due to the expression of ACE2 in the heart. There are other cancers and certain blood disorders that also increase it. So if you have cancer, your already immune system is stressed, and if now you become COVID-19 infected, your chance of surviving the infection are markedly reduced. The other is immune system. If you have an organ transplant, if you have HIV, if you're using a long-term use of a hormone that weakens your system, all these contribute to the disease. And the last is the risk factor increases dramatically if you have chronic kidney disease. And the reason for this, again, will become apparent at the end of my talk. So there are I could come up with 8 unanswered questions as of today. The first question, why are preteens and teens less susceptible to COVID-19 than the elderly? Could it be due to differences in innate immunity, physiology, or anatomy? And regarding the latter, the pediatric airway is smaller in diameter, shorter in length in adults, in those children than adults. Is that the reason? And my second talk, I'm going to focus on innate and acquired immunity. And a person who's infected in the first 24 hours is 100% due to innate immunity. So those people who asymptomatic, who never are able to show symptoms, their innate immunity is really remarkable. And so that's a whole area of investigation. What is unique about innate immunity in preteens and teens compared to the elderly? Another unanswered question is, why are hospitals doing a poor job preventing deaths for those who are 50 years or older? If you're 30 years old and you end up going to the hospital, you have a good chance of surviving and coming out of the hospital. If you're 85 years old and you and you go into the hospital, it's going to be really hard to make it out of the hospital. Why is pathology more severe in COVID-19 infected Blacks, Hispanic, and Native Americans compared to Caucasians and Asians? Again, we don't know the answer to that. Starting to make some discoveries, I think it's probably going to turn out to that there's single nucleotide polymorphisms or small mutations in critical genes that controlling ethnic susceptibility. Now, as I told you, the COVID-19 binds to ACE2, and then a tryptic-like protease at the surface of the epithelial cell activates it. And so the question is are there other tryptic proteases that also function to do this? And one of those proteases, one that my lab cloned, we were part of the Human Genome Consortium, and we identified this tryptase locus, and one of those tryptase is interesting, expressed in human epithelial cells. So the question is, does that tryptase epsilon, activate COVID-19? What is the mechanism of the cytokine-chemokine storm that occurs in those COVID-19 infected humans who require hospitalization? Is this storm due to mast cells, as I believe? What is causing the internal blood clots in COVID-19 infections? Is ACE2 the only receptor on the surface of human epithelial cell that recognizes COVID-19? And other than the lung, what other organs in the human body play critical roles in the pathogenesis of COPD? And it's these last two questions that are raised become quite pertinent to this data. Now, the Human Genome Consortium, what they did is they looked at where all 30,000 genes were expressed. In this group here, Fagerberg and co-workers, they looked at the human messenger RNAs in 27 different tissues in 95 different individuals, determined the tissue-specific expression of every gene, including ACE. Now, we know that people who are infected with COVID-19 have severe a lung problem, and the belief was that that was due to the virus binding to ACE2 and the lungs. But when you look at the expression of ACE2, it's not expressed in the lung at all, or very small amount. It's actually expressed in the small intestine, the kidney, the heart, and those tissues. So this data leads to the possibility that so many people asymptomatic because they breathe in the COVID-19, it gets into the lung, but it stays only in the lung. And there's so little ACE2 expression in the lung that the virus cannot grow and replicate. But in those people who develop severe disease, the virus is able to escape the lung, go throughout the body. If it gets into the small intestine, that's where ACE is expressed. It affects the epithelial cells, not in the lung, but in the small intestine. And the virus goes like wildfire. And indeed, this data, the expression data explains a lot. We know that about 30% of COVID infected people develop diarrhea, and that suggests the reason for that is because the virus is affecting the small intestine. We know that chronic kidney disease is a major risk factor, and note that ACE2 is expressed in the kidney. We know that heart is another problem area, and again, the heart is expressed there. So as of presently, February 2021, these are 7 areas that I identified which are unexplored or poorly explored areas. So what's the importance of the small intestine in dictating viral loads in COVID-19 infected human? It hasn't been evaluated. I believe that it's going to be the major area for where the virus is replicating. What is the importance of innate immunity in early stage of infection? And as I said, there are many people who are asymptomatic, and that is they have an innate immunity difference from those who are susceptible. They're able to combat the infection. What is the mechanism that occurs? We're going to talk a little bit about that in my second talk. What is the importance of mast cells and tryptase in the cytokine storm that occurs in COVID-19 people who require hospitalization? I'm going to talk a little bit more about that in my follow-up lecture. Why are the COVID-19 affected people developing blood clots? Now, there is a mechanism in the endothelial cells, epithelial cells, that controls blood clot, and it's dictated by the proteoglycan called cytokine-4. Is that the reason why the dysregulation of cytokine-4 is causing blood clots? Now, I told you that you activate COVID-19 by a particular tryptase, TMPRSS22, but there are actually a large number of tryptic families. And of those tryptases, do any of them fall as alternate ways to activate COVID-19? I told you that there's different susceptibility to pathogen is genetic dependent, and Afro-Americans, for example, are much more susceptible than Asian Americans. And is that due to mutations in the ACE2 gene, the TMPH gene, or some other gene that participates in the pathogenesis? And the last point, does COVID-19 induce harmful Ig responses in some affected people? So most of you who listen to this talk probably already had your first slide. And what was the one question that they asked you before they would give you the vaccine? Are you allergic? Do you tend to develop allergic reactions to antigens? And if you do, they wouldn't give you the vaccine. And I'm going to go into this in the second presentation. And the goal of the vaccine approach is to generate IgG antibody, but not IgE. If you generate IgE, you could die of systemic anaphylaxis. And that's why they asked you when you first got your slot, do you tend to become allergic to allergens and other compounds? So now I'd like to spend it on my last slide here, talk about some of the things, these closing comments. So the coronavirus, SARS, MERS, and COVID-19, they all affect the lung. They appeared in 2003, 2012, and 2019. And that's a deadly coronavirus every 7 to 9 years. And regrettably, it's only a matter of time before the next coronavirus or some other deadly pathogen appears. Now, coronavirus already has killed 506,000 Americans, and that's a horrible statistic. But its mortality is 3%, which is much lower than MERS, which has a mortality rate of 35%, or Ebola, which mortality rate approaches 90%. The bottom line, it could have been a lot worse. The problem is the leadership of the World Health Organization, the Center for Disease Control in the United States, and similar health agencies in other countries did not adequately appreciate the dangers of a pandemic in a world where an infected person could hop on an airplane and spread the pathogen to those in distant countries in a single day. The one country that understood this was South Korea, and that's why South Korea has very few deaths. Now, once COVID-19 was identified in China, the United States had 2 months to get prepared for the coming storm. And regrettably, little was done by the Trump administration during that time period. Mistakes were made. The failure to implement a mask policy, the failure of a faulty test that cost the lives of so many people. If you use the South Korea example, we probably lost 500,000 because of those mistakes. Now, moving ahead, Ebola was identified 47 years ago. And outbreaks of this terrible virus occurred in Africa every couple of years. There was an outbreak in the Congo in 2018, and there was another outbreak in 2020. Despite its greater danger, America still isn't prepared to deal with Ebola if it gets established here. It just takes one or two people in the Congo to get on an airplane to come to New York City to bring Ebola, and we start getting a huge number of deaths. Now, hopefully, the world's leaders are now aware of the steps that need to be taken shortly after the next coronavirus appears somewhere in the world. Aggressive, proactive approaches must be taken early on to minimize the chance of another pandemic in the United States. And here, we all believe America does everything great, but we did a terrible job. And the country that did it right was South Korea. And we have to use South Korea's example moving forward. Well, that's the end of my talk. And if you have any questions, feel free to send me an email. I gave you my email address. Thank you for listening to this presentation.